concluded. Dr. Xu, please go ahead. I'm sorry, we're experiencing technical difficulties. Please give us 1 second.
Oh, is this better now?
Oh, yes, Dr. Xu, now we can hear you. Please go ahead.
I'm sorry, it was muted before. Thank you very much, Christy. Good morning and good afternoon, everybody. It's my great pleasure to have the opportunity to share our research with all of you over the genome webinar. If my voice is low, please just type in the chat and I can notice that and raise my voice. And my special thanks for Thermo Fisher Scientific for sponsoring this 3 session of new frontiers of liquid biopsy research. Before I start today's talk, just a quick disclosure. This data presented here is the property of USC, and no USC content may be used without explicit permission from our institution. In the previous 2 sessions of this series, Dr. Bellocillo and Dr. Bramlett had introduced their work on adding CMVs and fusions to a long cfDNA assay. And later, Dr. Guagliata had shown his lab's research on liquid biopsy with Oncomine pan-cancer cell-free assays. So follow up with their 2 previous sessions. Today, I will talk about our experience in our lab with how we implemented the Oncomine cell-free DNA breast research assay V2 into our existing single-cell analysis platform, and how we integrated genomic analysis and assessment of analytic variable in single-cell circulating tumor cell, CTCs, and cell-free DNA from longitudinal blood samples. Here is the agenda for my talk. I will start with a brief introduction of our lab, our research focus and scope. Then the analytes of interests in today's talk, which is more than cell-free DNA. And then now in our lab, how we process the blood samples and tumor biopsies on different platforms, and then later merge the data and observations into a big comprehensive picture. After that, I will generally introduce the Blood Pack Consortium, in which we deeply collaborate with Thermo Fisher. And last but not the least, a showcase of our study of a metastatic ER-positive, HER2-negative breast cancer, with a focus on how Oncomine assay data is integrated with our copy number profiling and the morphological characterizations obtained on circulating tumor cells and solid biopsies from the same patient. Our lab, the Kuhn and Hicks lab in University of Southern California, is the cancer initiative of USC Michelson Center of Convergent Biosciences. We aim at integrating patient, model system, and high-content single-cell data to translate clinically observed correlations into a mechanistic understanding of the physical and biological underpinnings of cancer dynamics. The organizing framework of the physical dynamics of cancer focuses on the spatial distributions and the temporal evolution of the disease at the cellular, human, and population level. So around the topic today, I will focus more on workflow development and show 1 breast cancer case of our effort to develop the quantitative description with a multimodal liquid biopsy approach to understand the temporal evolution of the disease under the therapeutic pressure. This picture has been presented again and again in this session and also again today. Here I use the red color to highlight the advantages relative to our framework. The less invasive multi-sampling from 1 patient can provide temporal information for the disease evolution and the treatment response. then the liquid biopsy's nature of global representation of current disease status can be indicator of heterogeneity, and the potential to monitor both primary driver and resistant mutations can be achieved on both single-cell level and on analytes in plasmas. Here shows the components in bloodstream related to cancer liquid biopsy. CTC, which refers to circulating tumor cells in bloodstream. The origin of circulating tumor cells is not very clear yet. Although CTCs have been considered as the seed for cancer metastasis for a long time, it is yet unclear that which subclone or what population of primary or metastatic tumor has the ability to break into the blood vessel and survive for a long time. These cells circulating in blood with varying phenotypes, which is highlighted in the red box on the right bottom corner. Compared with cfDNA, which is highly fragmented shortened DNA fragments, the intact circulating tumor cell contains more information, which makes extensive downstream analysis like morphology, proteome, and a genome possible. But the challenges in detecting CTCs, therefore analyzing them, can make a very long list. On top of the list, the extreme low incidence rate makes finding single-digit number of CTCs among millions and billions of other blood cells very difficult. And also, we do not know what they all look like. So the proper identification and classification system is still not clear. So here comes to, so most of other CTC analysis approaches will use certain criteria to select or enrich CTC populations. In our lab, we use the no cell left behind approach to identify rare cancer cell with minimized cell loss. And we don't want any prejudgmental parameters bias our analysis when we don't have a proper classification system. So this approach will allow for a flexible cell identification process, wherein all nucleated cells in a sample are stained and imaged without prior cell population enrichment. Here comes the workflow of our high-definition single-cell analysis platform. Patient blood samples or bone marrow aspirates were collected from clinics, then shipped to our lab, and then processed within 24 hours after the time of draw. Plasmas will be firstly separated and frozen at -80 degrees for future use. Then red blood cells are lysed. Then the nucleated cells are plated as a monolayer on custom-made cell adhesion glass slide, followed by storage in a biorepository. 10 to 12 slides will be prepared for every tube of 10 ml blood. The slide will be thawed and processed with our standard 3-color assay when it is needed. Otherwise, the slides will be banked in -80 for a long term. So I will later use a couple of slides to introduce how the cells were characterized and analyzed with the principle of no cell left behind. Once the candidate cells is identified, it could be easily located again on the slide with the recorded coordinates. Therefore, the high-quality imaging, the targeted proteomics with imaging mass cytometry, and the downstream single-cell genomics will be performed on such said cell. So the blue line and the box here shows the new addition of ion-tolerant Oncomine cell-free Research Assay for evaluation of cfDNA as an expansion of our existing workflow. The integration of genomic analysis and assessment of analytic variable in single-cell CTCs and cfDNA, and add this information to the protein marker measured in CTCs, ensures us to have a more comprehensive understanding of the patients. So on each glass slide, average 3 million cells are plated as monolayer with a barcode on the side, which can direct us to our central data center. The cells on the slide are stained with DAPI, a pan-anti-cytokeratin antibody cocktail, and then an anti-CD45 antibody. Additionally, a third antibody will be incorporated for different study. For example, AR for prostate cancer study and the ER for breast cancer study. Images were taken with an automated fluorescent microscope, and CTCs were classified as cells that are CK positive, CD45 negative, and demonstrate other distinct morphological characteristics, such as cell and nuclear size, NC ratio, and subcellular localizations. So every cell will have an assigned coordinates for future reference. In the middle is the typical 10x scanning picture of one candidate cell, who is positive for CK signal shown in red, negative for CD45 signal shown in green, and DAPI in blue for DNA contents. As we mentioned, the tumor cells traveled in the bloodstream with various phenotypes. This direct analysis approach is designed to quantify and record morphometric parameters and protein expression levels in individual cells and cell clusters, and then to provide routine and clinically consistent access to any cell for downstream analysis. I want to specifically point out that these cells are not present in the absence of disease. We have done a lot of control experiments to make sure of that. And it is our lab's goal to identify their potential in disease. Next slide, please. Okay. Genomics. So once the candidate cell is identified with the coordinates, the micromanipulator is used to capture the cell and deposit it into a PCR tube for single-cell genomic analysis. Single-cell whole genome amplification is firstly done to bring up the genetic material amount. We are using whole genome shallow sequencing for copy number profiling. The key part is dividing aligned reads into 5,000 bins and counting GC-adjusted counts in these bins and calculate the ratio compared to the normal for each bin along the genome. So, two sample profiles shown on the bottom for demonstration. The left one, the flat one, is the CMV profile of a white blood cell, which has a normal diploid genome profile. And the right one is a typical single breast cancer cell, showing amplifications on chromosome 1q and the deletions on a lot of other chromosomes like 6p, 8p, etc. Since all of these information were obtained and calculated on a single cell level, which in theory all originated from 2 copies of genomes, so we are able to calculate the stepwise alterations here. Somatic copy number alterations in single cells always are numerical aberrations. The upper red line marks the 1 copy more on 1q, while the blue line on the bottom indicating all these deletions are exactly 1 copy less than diploid. This numerical stepwise alteration provides us a reference to calculate the tumor fraction of cfDNA, which I will explain more later. So for for cell-free DNAs, quantification with qubit assay and size profiling with bioanalyzer after extraction are a must-have QC steps before any downstream analysis. The amount of cfDNA is by itself a prognostic marker, and in our workflow, we are able to investigate the relation between CTC enumerations and cfDNA amount. Okay. So the same shallow whole genome sequencing strategy is used to generate the cfDNA CNV profiles. These two cfDNA profiles are from two different blood draws of one same patient. The upper line is a profile with higher fraction of tumor-derived DNA in total cfDNA, and the bottom line is a profile of a cfDNA sample with lower fraction of tumor-derived DNAs. For targeted sequencing on cfDNA, iron-tolerant Oncomine cell-free nucleic acid assay, together with Iron Chef and S5 system, is our choice for its high sensitivity and quick 2-day NGS workflow. The Oncomine cfNA workflow consists of 3 steps: sample preparation, including cell-free isolation, Oncomine library preparation, and a template preparation. In our lab, we're using Iron Chef system for the overnight templating. Then on the second day morning, the high-throughput semiconductor sequencing on Iron S5 sequencer. So the last step is using the integrated informatics solution for variance analysis on Iron Reporter. The core technology of Oncomine is an amplification-based assay that generates tagged DNA copies that allows to distinguish reads amplified from the same original DNA molecule and identify molecules containing variant. With deep enough sequencing, this assay enables detection of driver and resistant mutations from nucleic acid down to 0.1% limit of detection with a minimum 20 nanogram input. So here shows the portfolio of Oncomine cell-free assay families. We are currently using lung assay and the new pan-cancer assay. Both of them can generate single libraries for DNA and RNAs at the same time. But these two assays were previously covered in the 2 talks. I will focus on the breast Research Assay V2. Compared with the breast cfDNA assay, this Research Assay V2 has 2 more genes covered, CCND1 and FGFR1, and expanded coverage on TP53. Other than the SNVs, this assay can detect CNVs of CCND1, ERBB2, and FGFR1 on the same library. The lower limit of detection is the lowest allelic frequency of variants that can be detected with this assay. With a proper isolation, Tamia blood should be able to provide at least 20 nanograms cfDNA. The biological limit of detection of 20 nanogram genetic material is 0.1% with 6 mutation molecules presented in cfDNAs. Since we quantify every cfDNA sample we get, so we always reference back to the quantifications of cfDNA and keep the sample's limit in mind, especially when the frequencies of detected variant is on the borderline. So to evaluate the accuracy and the reliability before we implemented this workflow into our HD-SCA platform, we used 4 reference DNA control materials, which we called JFDI. We tested them before we merged these platforms together. So among the 4 reference samples, 3 of them contains same 115 engineered variants at 1%, 0.5%, and 0.1% frequencies. And among these 115 SNV candidates, 15 of them are covered by the design of breast cfDNA assay V2. We use this control sample because all these 115 15 SNV were covered by a pan-cancer assay. And we also verified the pan-cancer assay on this control samples. But here I just present what is covered by this breast research assay V2. So this table contains a list of 15 SNVs expected from 0.5% spiking control samples that each site can use as a reference. So this is our expectation from a typical 20 nanogram input. The expected numbers for 1% and 0.1% SNV control samples are also provided by Thermo Fisher for reference, which I will show side by side with our observed numbers later. The fourth reference samples is a genomic DNA background free of all these SNVs, which could be very informative for the environmental contamination control. And all these control samples were fragmented by sonication to mimic the size of cfDNA. Three routine quality control steps were strictly performed on each constricted library. I would strongly recommend fragment size profiling as step 2 shown here before templating the libraries onto ion chip. Good profiles with small peaks on smaller fragments, smaller than 200 side, and a big peak on bigger than 200 BP side. This kind of profile will provide more useful reads and a higher alignment ratio after sequencing. The first small peak is the primer dimers from the system, and the large second peak is what we want with our inserts inside. So the Thermo Fisher provides a standardized workflow and templates for Oncomine cell-free assays for consistent output from each operator in a lab or a research environment. So once we have our libraries constructed and QC'd, now we can use the Torrent Suite software, which is powered by Thermo Fisher Cloud, to plan the streamlines for ion chef templating and also sequencing run. The sequencing reads will be created to BAM files on the Torrent Suite and later automatically transferred to the Ion Reporter server for the downstream analysis and the reports. All the alignment, annotation, and the filtering will be done on the Ion Reporter with specific Oncomine plugin, which is designed to prioritize relevant variants and find the relevant new. Here, the next slide shows the typical Ion S5 run report. On the left side is the one generated on Torrent Suite, summarizing the quality of the run. And on the right side is the alignment report created by Ion Reporter. So for breast Research Assay V2, 4 million reads for each sample is expected in order to get detection limit down to 0.1%. Further down, we used the specific plugin for breast Research Assay V2 to call the variants. This report shown here is what you could expect when you click on the analysis result on Ion Reporter. But for this particular examples, because it contains 15 variants on the list, so it looks very busy and blurry. But in real examples, usually you wouldn't see such a long list. This table lists values for the sequencing depths, molecules counts for variants, and the frequencies counted based on the molecular tag. And of course, all these information is downloadable with one click. So finally, let's see. Okay, finally here is the table for the 15 SNVs in 1%, 0.5%, and 0.1% control samples detected with the breast Research Assay V2. So for every variant, the observed VAF is shown side by side with the expected VAF provided by Thermo Fisher. So we can see most of our observed value are very close to the expected value, which means in our lab, when we follow the standardized workflows and templates, we can consistently obtain reproducible results down to a limit of detection at 0.1%. So we have done this experiment repeatedly, twice, and not only with breast research assay, but also with pan-cancer assay, to make sure all these results are accurate and reproducible. Now we can move, we can implement the data, implement this Oncomine assay into our HD-SCA platform. So they will work together, implemented together as a standardized workflow for our metastatic breast cancer study in BloodPAC Consortium, Project 007. BloodPAC. The Blood Profiling Atlas in Cancer Consortium was launched on October 17, 2016. To accelerate the development and validation of liquid biopsy assays to improve the outcomes of patients with cancer. To do so, BloodPAC is developing a collaborative infrastructure that enables sharing of information between stakeholders in industry, academia, and regulatory agencies. Project 007 of BloodPAC is supported by Breast Cancer Research Foundation, and we are working closely with these listed partners in this project. This project is collecting and analyzing longitudinal blood samples from ER-positive metastatic breast cancer patients for a multi-platform, multi-analyte blood profiling analysis. Here is the design of the project. For each blood draw, total 40 ml blood will be split into 4 tubes, and each tube will be sent to one participating lab. So at least 3 blood draws will be collected from one patient. Up to 12 study-related blood draws will occur if there is a disease progression or change in treatment. So our lab will use HD-SCA plus Oncomine Breast Cancer Assay V2 to analyze the first blood tube, BCT1, and we commit to share data on the HD-SCA on CTC, morphology, genomics, and proteomics with simultaneous genomics on over 100 patients with breast cancers. So here is next slide. So here is one poster case showing what our data package will look like for one patient. So on the very top is the clinical information. Right down there is the immunofluorescence expression of estrogen receptor on HD-SCA analysis. We are able to show the ER expression level on single-cell CTCs over time, then correlate the single-cell genomic information with the cell's morphology. For some patients, like this one, we luckily have tissue biopsies, so we are able to compare the tissue genomics with single-cell genomics. For cell-free DNA, the whole genome CNV will be profiled and compared with single-cell profiles to estimate the tumor fraction in cfDNA, while SNVs will be profiled with Thermo Fisher Oncomine Breast Research Assay V2. On the right side shows our result of targeted proteomics using imaging mass cytometry with 40-plex imaging through laser ablation and CyTOF. So now I will focus on details of this poster patient. Let's know more about her. This was ER-positive, HER2-negative metastatic breast cancer diagnosed in 2012. Here are the MRI results at time of diagnosis in 2012. So MRI revealed a tumor in the right breast as well as metastases in the bone together with compression fracture of the spine. The next slide. Here lays her treatment history and sampling history. So in the past 6 years, she underwent 8 different treatments. The yellow colors here are hormonal therapy, and the dark pink blocks are chemotherapy, and the light gray blocks are the 2 targeted therapy she ever used. Then the short upward lines above the ears are the blood samplings. In the past 4 years, we have collected 17 blood draws starting at her enrollment in our study in October 2013. She was biopsied at 2 time points on 3 different tissues. In 2012, when she was first diagnosed, both primary breast site and the metastatic bone site were biopsied. And later in 2014, another new metastatic site on liver was further biopsied. So we collected 17 blood draws, and then all 17 blood draws went through our standard HD-SCA platform for CTC characterization and enumeration. The gray bars here are the concentrations of CTCs for each blood draw. The high cutoff of the enumeration set at 3,000 CTCs per ml blood. Among the 17 blood draws, 4 of them reached that cutoff, and 5 draws around 2015 have lower than 10 CTC counts in every ml blood. The expression biomarker CA2729 was tested over time, and here overlaid on this graph as orange dots. We can clearly see that the concentration of CTCs was rising and falling at the same time as her tumor markers, so indicating that in her case, the CTC enumeration has the sample predictive power as CA2729. So for the single-cell genomic analysis on these 17 blood draws, we totally picked over 700 CTCs and did single-cell genomic analysis on all of these 700 CTCs. A total 4 subclones were identified, and all of them have evolved from one single clone. On the left side, here listed the represents of each clone. Morphology, the cell morphology in the middle, and the copy number alterations, the chromosome around the cell as a circle. So we propose that subclone 1 to be the earliest CTC subclone based on the observation that it exhibits the least CMV. Then based on subclone 1, 2 more deletions in chromosome 8p and chromosome 18p observed. Then from there, 2 more amplifications in chromosome 6p and 11p observed in clone 3. Clone 1, 2, 3, these 3 subclones, were found ever since the very first blood draw in 2013 through the 17th. While the clone 4 was only found in the very last 2 blood draw in 2017. We will have one slide dedicated to clone 4 later. CMVs of these cells indicate the evolution of the disease, manifested in the increasing numbers of amplifications and deletions. But despite their genomic differences, the cells from these clones cannot be distinguished by immunofluorescence or morphometric analysis. So next, we compared 15 single-cell CTCs from blood draw 1, shown in this heat map, with the 3 tissue samples. Then the way to read this heat map on the left side is From top to bottom is the chromosome number from 1 to X and Y. Then from left to right, every column is one sample. The first left three are tissue samples, the primary breast, the metastatic bone, and metastatic liver. Then 150 CTCs on the right side. The red color indicates amplifications in that region, while the blue color is the deletion. The three arrows on the left side point to the additional alterations found in that color code subclones. We can see all 150 cells from that blood draw clustered clean and clear into 3 clones. Orange color for clone 1, green for clone 2, and the blue for clone 3. And among the 150 cells, the green color clone 2, subclone 2 is the majority population in this blood draw. So now on the right side, we found the genomic profiles of both primary breast tumor and bone metastasis only contains cells that resembled clone 1. But the liver metastasis, which was biopsied in 2014, late 2014, 2 years after the biopsy of bone and breast. So in this liver metastasis, solely of cells from clone 2. It is remarkable to mention that clone 2, subclone 2, was detected in our very first blood draw, which was taken in 2013, almost 2 years, almost more than 2 years before the detection of the liver metastasis. So this suggests that we can detect new genetic aberrations in CTCs before they form a detectable tumor mass. None of the tissue examined contains cells corresponding to subclone 3 or 4. But it remains open whether a new metastatic site will be detected in the future containing 3 or 4 clones. So this patient remained a very stable genomic, consists of only 3 subclones over 4 years, until the second chemotherapy treatment. The clone 4 was discovered for the first time with a predominant proportion in draw 16. And at that time, she was progressing on both hormonal and chemotherapy. I didn't put the picture here, but when we test the ploidy of CTCs with fish, we found tetraploid CTCs for the first time at the same time point and proportion of clone 4. So this observation hinting that subclone 4 might have been created by whole genome duplication of clone 2. It suggests that clone evolution could happen by either chromosome-specific copy number changes or whole genome duplication, which of course possibly due to a failed cell division. Then let's put all the 700 single cells from 17 time points together in one big picture. So all analyzed CTCs genetically match with one of the 4 subclones. The abundance of the clones varies depending on the treatment or the therapy response. For the cell-free DNAs, as we mentioned earlier, since all aberrations on single-cell levels are stepwise, so it provides an opportunity for us to calculate the tumor-derived DNA fraction in cfDNA based on the amplitude of amplifications and deletions of the CMV profile. So what we have done is we calculate the ctDNA. We calculate the ctDNA percentage, the fraction, with comparison of the cfDNA CMV with the single-cell CMV, and we found that time points with elevated CTC concentrations also contain larger fractions of ctDNA, and the levels are positively correlated. And also, CNV profiles of cfDNA represent the aggregate of CTC clones. For example, for the draw5, when the subclone 2 is the dominant CTC clone, the cfDNA profile will show the clone 2 profile. So cfDNA CNV profiles reflects the very most abundant CTC clones at that time point. So here is the overview for all our plasma samples. This is the CNV result. And what about SNVs? So here on the bottom is the treatment information and the timeline. We try to understand why the only treatment that the patient responds to is chemotherapy after relapse on letrozole. And none of the hormonal drugs appears to have a sufficient effect on keeping her tumor markers in check. In our single-cell morphology characterization, although we don't see correlation between ER expression and a specific CNV subclone, we did notice cells expressing ER decreases to 10% under the ER deprivation therapy with fulvestrant. But we cannot find a good explanations with CNVs, but we have like a feeling that the ESR1 resistant mutation might be the reason. So we started to detect that mutation on both single cells and plasma samples. We didn't collect the plasmas until the blood draw 5. With the plasma samples from draw 5 to 17, we use this Oncomine Breast Research Assay V2 for the SNV analysis. But due to the limit of our plasma samples, most of our analysis were done on 1 ml plasma extraction. That's why I highlighted the draw 9 and 10 in red with the total cfDNA amount around 8 nanogram and 6 nanogram as the Oncomine input. So when we have the result for the blood draw 9, the result shows 0.1% VAF of TP53, but we just keep it in mind. This mutation is on borderline, and theoretically with 8 nanogram input, this result is suspicious. But we repeatedly see this mutations in the later plasma samples as well. Then here is the table for the variant results obtained on this platform. We found ESR1 mutations in almost all the blood draws, starting from the draw 5, the first plasma samples we tested. So this result indicates the ESR1 resistant mutation arising and responsible to the failure of fulvestrant treatment. And as we said, at the same time, we are trying to find the ESR1 mutation on single cell levels. And all these the arrow-pointed mutations were confirmed on our single cell CTCs with Sanger sequencing. We found the ESR1 mutated cells as early as in draw 3. But we don't have the plasma for draw 3, so we don't have a result on plasma from draw 3. But on the single cell levels, that mutation, that particular ESR1 mutation, was detected as early as in draw 3, which is the first blood draw in 2014, still in fulvestrant treatment. But after that, she was no longer respond to that treatment. So all these results taken together I forgot to put the biopsy result here. We also detected this ESR1 mutation in liver biopsies, in liver metastatic biopsies, but not in bone and the primary breast biopsies. So all these information taken together supported this ESR1 mutation is induced by hormonal treatment, and is the reason for she's no longer responded to the later hormonal treatment. A big surprise from Oncomine result is we found both PIK3CA and TP53 mutations in draw 16 and 17. This combination wasn't there before in the earlier plasma samples. So compared with her treatment history, draw 16 and 17, both of them were sampled in 2017. This suggested that the palbociclib, this selective inhibitor of the cyclin-dependent kinases CDK4 and CDK7, probably is the reason. This drug probably induced the PIK3CA resistant mutation, and then also responsible for the uprise of the subclone 4. We are currently testing this hypothesis, and we don't have a solid conclusion yet, but we would like very much to know what is happening on single cell level and how we correlate that with the Oncomine result, and to explain the uprising of clone 4. So the summary of this poster case, we find the high concordance among expression clinical biomarker, tumor fractions in cfDNAs, and CTC concentrations. And also the genomic information of liquid biopsy reflects the nature of tissue biopsies. And also the treatment caused ER resistance in tumor cells, and which enriched in both metastatic liver sites and the liquid biopsies after the treatment. So taken together, this patient example is a proof of concept of how liquid biopsy can guide treatment decisions in the future. So then we go to the very last slide of today's talk. Liquid biopsy and the Oncomine assay. Using liquid biopsy to capture the intra-tumor genetic heterogeneity and comparative mutation analysis on CTCs and the analyze on cell-free nucleic acids provides a unique opportunity to understand the disease evolution and the treatment response. But in the future, in the era of precision oncology, which of these two approaches will become the eventual gold standard for managing the patients is remaining open. I think in my mind, they are partners, not real rivals, especially when we want to know comprehensively of the disease. And all of our work was supported by Breast Cancer Research Foundation, and great thanks for all our supporters and funding resource and the partners. And great thanks for our cancer initiative and USC Michelson Convergent Biosciences. This is a very interdisciplinary work, requires a lot of input from every perspective, and we are lucky to have a lot of genius working on our team. And thank you very much for your time. Have a good day.
Thank you, Dr. Xu. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. Okay, so we'll now start the Q&A. Dr. Xu, it seems like most liquid biopsy CTC studies are looking at later stage cancers. So how early in cancer can CTCs or ctDNA be found in blood, and is it different in breast cancer versus other types of cancer?
In our lab, we do have a population of early stage breast cancers. We found average between 5 to 10 CTCs per ml blood sample in early stage patients, but not all of them have this typical cancer candidate morphology. And also for genomics, all these single cells don't really have a distinct clonal genomic alteration. So it remains unclear if those cells, cytokeratin positive cells identified on our platform, are indeed derived from the primary tumor site. But we do see cytokeratin positive cells in early stage patients.
What's the concordance of mutation calls between a patient's tissue and biopsy before and after treatment?
Excuse me, I didn't hear, it's some noise. I didn't hear the question clearly.
Oh, I'm sorry. Can you hear me now?
Oh, yes, please.
Okay. What is the concordance of mutation calls between a patient's tissue and liquid biopsy before and after treatment?
That's a very good question. So for liquid excuse me, you're asking the tissue biopsies or liquid biopsies? I didn't
tissue, the concordance of mutation calls between the tissue and the liquid biopsy.
Yes. So for the tissue biopsies, because we don't really have a chance to get a lot of tissue biopsies from the patient, and sometimes, most of the times, it just come in when they primary diagnosed. And for this case, like we found the new ESR1 resistant mutations in livers, but not the very early primary breast site and the metastatic bone site at the diagnosis time. So I would say for the result on the blood draw 1 and 2, which we never found any ESR1 mutations, there's a high concordance between the cfDNA and between the mutation detections on solid tumors and the liquid biopsy tumors. But as I said, the only late biopsies we have is the liver. We don't have any more other biopsies from this patient after 2014, and further 6 treatments after that. So for the only time points we have is the 2 times gap. So for those 3 samples and those 3 blood draws, they showed very high concordance.
Early on in your talk, you said that about 10 milliliters of blood equals 10 nanograms of cfDNA. Is this dependent on the patient's cancer status?
Yes and no. We do find a high cfDNA amount in normal blood donors as well. But in general, a big population, like 80% late-stage cancer patients will have higher average cfDNA amounts. There are outliers in every group, though.
What if there's a conflict that occurs between the results of the liquid biopsy and what you see on the anatomical slide?
You mean the CyTOF slide?
Yes.
I'm sorry, I don't have a complete information of CyTOF results because my expertise is more on genomic side. So I'm sorry, it's hard for me to answer that question correctly, but I'm happy to talk over the emails and get more details on that.
Okay, great. Given the information that you can get from the Oncomine Breast cfDNA assay, what value do you think it could provide for the treatment of patients?
Well, first of all, this kit is still for research use only, but as we shown here for this particular patient, if we have the chance to test her plasma samples when we get the blood at that time, then we will be able to tell her the ESR1 mutation is showing up in her blood. And then we would advise her clinics, her physicians to skip hormonal treatment for her later 2 years.
Are there any guidelines to follow for the cfDNA and ctDNA test validation?
We are establishing a generic protocol in the Blood Pack Consortium, and hopefully we will have something coming up soon. But that's one of our major objectives for the whole Blood Pack Consortium.
Do you have any idea of the relative cost of the tests and how frequently you would expect a patient to be sampled?
Are we talking about HD-CTC or the Oncomine assay? The Oncomine assay, right?
Yes.
Okay. So for the Oncomine assay, I would say how frequently the patient be sampled depending on the frequency of her doctor visits and her disease progression. So if her disease remained good and no progression observed, then I would say maybe half a year. Especially for breast cancer, we don't really expect a lot of resistant mutations showing up like as fast as lung cancer. And for the cost, it is about $200 to $250 for the biochemistry reactions. But of course, the machines comes with other as another price. But for once you have the machine, every run for every sample with this assay is about $200 to $250.
What's a better indication that a tumor is returning? Is it SNVs in certain genes or hotspots, or is it CNVs?
Depending what samples you're using, because in most clinical labs, plasmas and cfDNAs is the most available samples. And in that case, SNV testing is more sensitive than CNV testing. Because, for example, for our when we compare our cfDNAs, CNVs with the single-cell CNVs, we have a limit of 5% tumor-derived fractions detection limit in cfDNAs. So lower than that fraction, lower than 5% tumor-derived DNA fractions in cfDNA, it is very hard to tell exact how much in the cfDNA pool. However, when you use the Oncomine assays with the molecular tagging, it is able to push down to 0.1%. At least with a general 10 nanogram input, 1% is not a problem.
Great. Thank you, Dr. Xu. And that's all the time we have for today. If we didn't have time to get to your question, we will try to follow up with our expert. We'd like to thank Liya Xu, and special thanks to Thermo Fisher Scientific for having sponsored this very informative three-part series. If you missed parts 1 or 2, you can find them posted to the webinar library on genomeweb.com. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.